Okay, welcome to the world of physiology. This is episode 4, membrane transport for medical students, for undergraduate medical students. Here we are going to discuss the resting membrane potential and the action potential, the depolarization phase, repolarization phase, and hyperpolarization phase, action potential propagation, axonal transport in the form of anterograde and retrograde. Yeah. So membrane potential. The resting membrane potential is the steady transmembrane potential for a cell that is not producing an electric signal. It is determined by the passive movement of several ions. All cells have a negative intracellular potential when it is measured by electrode. Okay, you see, the inside is negatively charged and the outside is positively charged under resting membrane potential. And when it is measured using microelectrode, it depicts about minus 74 millivolt. So the position of this resting membrane potential is ionic gradient between ECF and ICF, inside more negative and outside more positive. So this amounts from minus 50 to minus 100 millivolt, an average minus 17 millivolt. So causes, as I told you, diffusion of ions, selective permeability of plasma membrane, and electric attraction of negative and positive ions. Nerve cells have a low threshold for excitation. This is the extracellular side, and this is the type of the cytoplasmic side. As you see it, the cytoplasm is negatively charged and the extracellular part is positively charged. So excitable tissues are nerve and muscles. We will discuss later on in each and every part. So initiation of the action potential. What initiates the action potential? So the threshold for initiation of an action potential is about minus 65 millivolt. Okay, so the direction of propagation is in one direction. Okay, there is a principle of all or nothing principle. This all or nothing principle is if the threshold, if the signal, or if the stimulus reaches to a threshold level, it is the action potential is in occur all time. But if it doesn't reach the threshold level, action potential couldn't occur. So this is the all or nothing principles of action potential. So propagation of action in here in order is A, resting membrane, then action is inside, then B is occurring, then C like this. Okay, how it is propagated. So this is a neuron. Okay, this has a soma and a dendrites that receive impulses, where it has an axonal hillock where action potential is generated. And here is the axon. Here it has an axonema and endoneuranium. So here north of rambar, it has its own structure, and the collateral, then presynaptic terminal or button or axon terminal will occur, and it will communicate with the second neuron. Okay, so electrical transmission will occur in these forms. Here vesicles are formed, co-chemical transmission or neurotransmitters are released in the form of exocytosis in the synaptic cleft, then the electric transmitted into post-synaptic membrane. Okay, so the neurons have four important zones, as I told you. One is a receptor or a dendrite zone. The second is the site where propagation of action potentials are generated, and an axon process that transmits propagated impulse to the nerve endings. Finally, the nerve endings. Okay, this is important. This is how action potential is going to occur through the action potential in times in milliseconds and the resting potential. Here is the resting membrane. Then when depolarization phase occurs, sodium will enter into the cell, then it will cite a period of overshoot when it exceeds 0 millivolt. Okay, when the negativity is exceeded to the positive side, the overshoot will occur. Then the repolarization phase, when potassium is exited or be out of the cell, and the hyperpolarization phase will occur due to high efflux of substances below to the resting membrane potential. Okay, so activated gates like sodium could activate, and in activation gate resides, so sodium will be activated from minus 90 to plus 35 millivolts, then inactivated from plus 35 to minus millivolts, so the gate will be closed. So in the resting minus 90 millivolts, potassium will be all closed, and slow activation, potassium will leave the cell as a cell. Okay, so the depolarization release has changed the negativity and the positivity of a neuron. So the plasma membrane include like this. Then speed of depolarization will occur in such a fashion along the axon. So generation and conduction of action potentials. An action potential is a rapid or non changes in the membrane potential followed by a return to the resting membrane potential. Voltage dependent ion channels in the plasma membrane are the basis of action potentials. An action potential is propagated with the same shape and size along the entire length of an axon. Action potential are usually initiated at the initial segment of an axon, as I told you, in the axon hillock. The action potential is the basis of a signal carrying ability of nerve cells, and the patterns of coded conducted action potential encoded the information conveyed by the nerve cells. So this is how action potential occur. Okay, here is times, and here is in millivolts, and times in milliseconds. Okay, depolarization is a change of the charge difference across the plasma membrane, making the charge inside of the cell less negative and the outside of the plasma membrane less positive. As you see figure B, during the depolarization phase, the membrane potential changes from approximately minus 85 millivolts to approximately plus 20 millivolts, as we see it here. Okay, during the repolarization phase of the resting membrane potential, the inside of the plasma membrane changes in charges from approximately plus 20 millivolts to minus 85 millivolts. This is the repolarization phase of the action potential. Here it is. So the strength of a stimulus is matter in order to initiate an action potential. So a stimulus threshold is the weakest stimulus that can be reliably detected. So there is tension and there is increase of stimulus in strength. So there is subthreshold stimulus, no motor units will be responded in the muscle. Okay, threshold stimulus or one motor unit or respond will occur, but submaximal stimulus increase the number of motor units respond, then firing of action potential will increase, but in maximal stimulus all motor units will be responded, and in the supramaximal stimulus all motor units will be responded. Also there is a firing of action potential. So phases: first, repolarization, inside plasma membrane becomes less negative, and repolarization where a return of resting membrane potential. All or none principle, as I described earlier, is like a camera flash system. Propagation, spread from one location to another, and the frequency, number of action potential produced per unit of time. There is also a refractory period. During much of the action potential, the cell is completely refractory to further stimulation. When a cell is refractory, it is unable to fire a second action potential, no matter how strong it is stimulated. This unresponsive state is called the absolute refractory period. During the later parts of the action potential, the cell is able to fire a second action potential, but a stronger than normal stimulus is required. This period is called the relatively refractory period. So diagrammatically, as it is here, absolute refractory period here. In this case, no matter stimulus is initiated, as no action potential could occur, but in the refractory period, action potential could be generated. Okay. So synaptic transmission usually occur via channels of neurotransmitters. This is action potential coming from first neuron, then action potential 1 will release, then the synaptic or the dendrites will receive the impulse and it will propagate, and the current will leave and the axon hillock and it will be propagated. So integration of postsynaptic potential occur in the dendrite and soma. So excitatory postsynaptic potential 1 and excitatory postsynaptic potential 2 here in the membrane potential at axon hillock when action potential 1 is occurring and action potential 2 is occurring, so they will summate. Okay, so action potential, excitatory postsynaptic potential 1 and excitatory postsynaptic 2, okay, in such a way, so they will summate. So there is also graded potentials. Depolarization or hyperpolarization can occur. Increase with an increase in the strength of the stimulus, propagate in a decremental manner in such a case of graded potential. Since depolarization can lead to the excitation and activation of a neuron, they are commonly called excitatory postsynaptic potentials. In contrast, hyperpolarization of the membrane prevents the cell from becoming activated and are called inhibitory postsynaptic potentials. So these membrane potential changes are caused by the influx or efflux of specific ions. So graded potential, excitatory postsynaptic potential, and inhibitory postsynaptic potential. The first one is the membrane potential in millivolts versus the time in milliseconds. So this is the excitatory postsynaptic potential occur at minus 16 millivolts. But here below minus 60 millivolts there is an inhibitory postsynaptic potential occurring. Okay, so excitatory and inhibitory postsynaptic potentials. So stimulus or process and information coding will occur because first there will be a sensor. So there is a sensor, then there will be information coding, then there is propagation in the presynaptic neuron, then the recording will occur, then it will be synapse in the postsynaptic neuron, there will be a recording, and the postsynaptic neuron will propagate in such fashion. So this is how time and sensor potential will fire. This is the threshold how they are firing, okay, in each part, okay, the postsynaptic potential and threshold. So axon transport, axonal transport can be anterograde. This is the movement of materials in from the soma or cell body towards the axon terminal. It can be fast or slow depending on substances moved. In the retrograde one, the movement of material is from the axon terminal back to the cell body. So microtubules are used for vesicle transport in the fast axonal transport. These are the cell body, how anterograde transport is occurring like this. Microtubules are involved. This is the terminal or the synapse. This is the axon. So in the retrograde transport, this is occurring from the terminal to the soma. Okay, so microtubules oriented with the plus end sign or distal towards the synapse. Kinesin powers anterograde transport through kinesin to the synapse. Cytoplasmidynin powers retrograde transport to the cell body. Use ATP hydrolysis to work towards the minus end. This is how it is moving from minus end to plus end by transport vesicles by kinesin. Okay, like this, kinesin working on microtubules. Okay. So retrograde transport return used synaptic vesicles and other materials to the soma. Some pathogens also exploit this process to invade the CNS. They enter the distal tips of an axon and travel to the soma by retrograde transport. Okay. So by the way, why do cancer cells kill? Can you explain? This is all for today. I will come with the next lecture with a specific time. I will come soon. Please don't forget to subscribe more audio lectures of physiology. Thank you in advance.